consumable. These are edible substances which contains some important compounds. Those compounds are very important for our body functions, for maintaining our health, for preventing different diseases, for maintaining different diseases. That means the food contains some substances which are, as I said, essential, and we call them nutrients. The food contains nutrients, and these nutrients are very essential for us. That's why we can process this and take out the essential items, nutrients, and eliminate non-essential items, wastes. Okay, that is not required, so we will excrete it in the form of feces, fecal matter. So, food contains nutrients. We divide these nutrients into different categories. What are these? We call them proximate principles of food. And these nutrients there are 6 proximate principles. What are these? Fat, protein, carbohydrates, vitamins, minerals, and the last one? Water. Okay. If we get this, they will be able to serve all my body needs. Our body will be able to function properly. It will be able to maintain the body functions. For functioning of the cells, for functioning of the body, we will need energy. For this purpose, to get energy, so there are some nutrients. The nutrients which can provide this energy, they are called calorigenic food. They are called calorigenic. Can vitamin provide calorie? They are not calorigenic. Can minerals provide energy? Can water provide energy? No. Who can provide? Yes. There are 3 calorigenic food. Calorigenic nutrients. I will not say food, we will say nutrients. Okay, these are the essential things that are present in food. So these calorigenic foods, what is their energy value? Means how much energy they can give per gram of these nutrients. These are called calorie equivalent. What are these? Calorie equivalent. What is the calorie equivalent of carbohydrate? 4 kilocalorie per gram. And of raw protein? Same. And of fat? 9 kilocalorie. 9 kilocalorie. So per gram, it can provide more than double. So I will not eat any other thing. I will eat only fat. Okay? I will only eat fat. Okay, you eat. So you have to eat less, then you will get more energy. Can you think of eating only oil? Can you think of eating only butter? No. Somebody may say, yes, I love butter. আমি তো এমনি খাই. One advertisement was there previously. You were kid that time. Moon Sen. She had this advertisement. She was the person. Amul butter. Advertisement of Amul butter. So that person, Moon Sen, she likes to eat butter. Okay? So she wants to survive depending on only butter. Okay, let's see. For energy, she will take only butter. What happens? What will happen? Oh, dude, it's much later. Before that. Are immediate effect, acute effect. Yes. First of all, she won't be able to digest all this fat. Some would be excreted. And in the stool, there will be a lot of fat. Slippery thing. It happens when liver function is deteriorated, pancreatic function is deteriorated, fat will not be digested. So whatever fat we are ingesting, that will be passing through stool. Since you are getting only fat, so there will not be any bulk of stool. Only small amount of there. So what you will be trying? But nothing will come out. Okay, so stool will be very less. Number one. Secondly, okay, some part is absorbed. So since there is no carbohydrate, there is no protein, only thing is fat. So we will metabolize it and ultimately to provide energy. How? Not you. How it can provide, how the fat absorbed as monoglyceride, diglyceride to some extent, and glycerol and fatty acids. So it has entered ultimately into the blood. Now it will be coming to liver first. So liver will distribute it throughout the body, through blood, and okay, you need energy, take, I have only fatty acids, take. So what it will be, what I will do with these fatty acids? I have to derive energy out of this. Okay. They said there will be beta-oxidation. So beta-oxidation, you studied beta-oxidation, I think. Huh? Not. The beta-oxidation is over, I think, by this time. Not. Okay. What is beta-oxidation? Why the term is given beta-oxidation? Say palmitic acid is one of the fatty acids. Palmitic acid is having 16 carbon. Carbon 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 16. Last carbon is 60 and it is having one carboxylic group. Last carbon is carboxylic group. Okay. So this is the first carbon from that side. Last one is first carbon. And then there will be another carbon, second carbon. And this carbon is called alpha. Who said? Alpha. You. Very good. And next to this? Beta. So oxidation will be, in beta carbon there are two hydrogen. Next to this, also actually all the carbons are having CH2, two hydrogen atom. So these hydrogen atoms are having energy. The electron of hydrogen atom is having energy, that energy. This energy will enter, it is happening in cytoplasm. Now it will be transferred to mitochondria, going there, and it will give its hydrogen atom to electron transport chain. And you know what then happens. Other day I have discussed. So there, the electron will be transferred. During this time, ATP will be, some energy will be during transferring of electron, some energy will be evolved, and this energy will be captured by ADP and inorganic phosphate, and they will be transferred into ATP. Now currency I got. Now I can spend. So in this way, I can generate ATP with this, I can perform. So in alpha and beta, so there will be oxidation. Removal of hydrogen means oxidation. So you will find repeated oxidation is there. Again water will be added. Again there will be OH and H. Again there will be the removal of hydrogen atom. So, and finally what will be there? This 2 carbon, the alpha and the last one, I mean the carboxylic group, they will be splitted in the form of acetyl coenzyme A. Now this acetyl coenzyme A will enter into mitochondrial TCA cycle. There it will be oxidized and produce 12 ATPs. Then it will provide ATP. By this time, what happened? When it is splitted, okay, I gave you, I am sure I am giving you one beaded chain. Having one bead means 1 carbon. So one chain having 16 beads. Now I told you, you do one thing, you split the beads 2 at a time. 2 at a time. How many times you can do it? 8 times, okay? How? Show me. Draw in your copy 18 beads. Draw? 16 beads. 16 beads, yeah. No, to gap. You will be splitting, no? What happened? Huh? No, she said 7 times. But you told 8 times. How come? How it has been reduced by 1? Can you do it 8 times? Is it possible? Yeah. There will be 7 times beta oxidation. Every time when beta oxidation is completed, means 2 bead is separated, 1 beta oxidation complete. Now you think this 14 carbon as a new fatty acid. Again it will be having alpha and beta. So again there will be beta oxidation. So 2 times, 3 times, 4 times, 5 times, 6 times, 7 times. And if it is uneven numbered fatty acid, say 17, so after last what will be 3 carbon acetyl coenzyme A. And lot of acetyl coenzyme you are getting. And all these acetyl coenzymes are not required for my required energy. Some acetyl coenzyme A are excess. What I will do with this as excess acetyl coenzyme A? I will convert it into ketone bodies. Acetoacetic acid, alpha-ketobutyric acid, acetone. So these are the ketone bodies. So that means production of ketone bodies, the process by which ketone bodies are formed, is called ketosis. See, I am a diabetic patient. Means I have deficiency of insulin. I am eating starch, carbohydrates, but I am unable to use it. My tissues are unable to use it since they are unable to enter into my cell. So they are there in the blood, but I cannot use them. The cell cannot use them. That's why blood levels of glucose is high. So cell has to depend on fatty acid. They have to depend on fatty acid or on protein. If I don't get sufficient protein, then my body protein will be utilized for energy purpose. I will become gradually thin. And if I use fat, there will be ketosis. Increased production of ketone bodies, and these are harmful for the body. So we'll try to eliminate this. So my urine will smell, good smell, because of acetone. Because of acetone. So if you get this smell, or if you find in biochemistry, you will be performing test for acetone. Can you name one test? Rotheras test. Any other test? So by that, you can identify whether your urine is having acetone. So somebody's blood, somebody's urine, you found it is positive. Means there is acetone. Normally it is not there. So immediately you can diagnose what is happening. Means in the body, fat metabolism is mostly, energy is coming from fat metabolism. Why body is utilizing fat? The person is eating carbohydrate. Even then, what is the problem? Carbohydrates cannot be utilized. That's why fat is eaten. That means the patient may be diabetic. So I will suspect diabetes. Another person is not diabetic. He is not diabetic. Okay? What is happening? He is fasting. He is starving. Somebody is starving. So no intake of carbohydrate. I am using this fat. These are fat deposited in adipose tissues. So lipase will break down them. So what will be formed? Fatty acid and glycerol, they will be transferred into the blood, and this fatty acid will be circulated through the, then we will utilize it. Because stored glycogen in liver and muscle, that was finished in the first few instances when there is low blood. So when you will be utilizing, there is no input of carbohydrate. Means blood levels of glucose is gradually falling. You are uptaking this, you are taking in, utilizing this. So gradually blood levels of glucose is falling. This hypoglycemia, this is called hypoglycemia. This will stimulate islets of Langerhans. Arey Baba, blood sugar is falling. Immediately you do something. Raise the blood sugar level. This person is not eating. Can you do anything? Yes, I can. How you can? Let us go for, in the liver, go for glycogenolysis. I know in the liver there is stored glycogen. So, this will be initiated. Activity of the glucagon will be increased. Insulin favors glycogenesis, production of glycogen, by utilizing excess glucose. Now glucose is not excess. So no question of glycogenesis. Now I need glucose, means I need breakdown of glycogen into glucose. So who can do it? Glucagon can do it. So alpha cell will liberate, hypoglycemia will stimulate alpha cells, it will produce, go to liver and it will stimulate the hepatocytes. अरे जितना भी तुम्हारा पास है उसको ब्रेक डाउन करो, ग्लूकोज को सर्कुलेशन में दे दो. Another thing it can do, gluconeogenesis. I need glucose. So it will, liver will do it. Gluconeogenesis. You know the terms, but you don't know the utility. When this process will be utilized. Now there is crisis for glucose. So now glucagon will stimulate gluconeogenesis. Means glucose production from non-carbohydrate substances. So from non-carbohydrate substance, glucose will be produced and will be given to circulation. Now this will be utilized. So how long it can go? Not much time. So after that, glycogen will be finished within that day. No more stored glycogen. No more raw material for gluconeogenesis. Then come. No other alternative. You utilize fatty acids. Then it started utilizing fatty acid. Consequence, ketone bodies. So this will come into the urine. Ketone bodies. So in case of fasting, in case of starvation, you may get. So either the person is diabetic, or the person, you didn't see the patient. You only say, I have seen the urine only. So by studying this, you can assess, the patient may be suffering from diabetes or fasting, or I am running for long period. Say a marathon runner is running. He is utilizing all his stored energy. Now he has to sustain his exercise. It is not high intensity exercise. You know, marathon is not a high intensity exercise. It's a low intensity but prolonged period. So in this condition, what will happen? After some time, fat will be utilized. That's why we call, if you want to reduce your fat, I got one. I got one. Another. I got two. So if they want to reduce their fat by doing exercise, what they have to do? Run for a longer period, so that time comes to utilize the fat. Then only the fat will be utilized. Or they has to exercise, high intensity exercise, sweat has to come. Unless you cannot reduce your weight. So if you want to reduce your weight, you have to do daily vigorous exercise. Then only you will be able to reduce your weight. So this is the third condition. Either just he has come after playing football, then gave you the urine, or after marathon run, then he gave the urine. So here you may find ketone bodies. Understood? See, I have debated long living this. So, food. My question was on food. Okay. So, foods are having nutrients. Their main function is to provide energy. Then, protein. Protein is this thing. Very important person. Very important person. Like the principal of a college, the teachers of a college. And there are some group D, group C persons. They are doing, somebody is doing dang, dang, dang. And somebody is taking file. Very simple job. Okay. This job can be done, if necessary, by the principal, by the teachers. If I want, I can do it. It's not so tough. I can bring the file. Simple circle. My teacher, they are teaching. If necessary, they can do that job. Can they, those who are, can they take the class? But they can do those job. They cannot do this job. That's why I am very important person. The protein is like that. I will not use the protein for that purpose. That purpose is energy generation. Unless it is needed, no group C, no group D employee came. But institute has to run. In that time, I will be doing that job as well as this job. If necessary, I can do. Protein can provide energy. But carbohydrate, fat, they cannot do the job of the proteins. So they are very vital for the body. Isn't it? So what vital functions they are performing? See, one example. My eyes. What is there in retina? Some pigment. What are they? I'm giving simple example. What is there in the retina? Some pigments are there, rod cell, cone cells. What they are having? Some pigments, rhodopsin, iodopsin. What are these opsins? Proteins. So, without protein, is it possible to form iodopsin? So, if protein is not there, will you be able to see somebody? You will not be having vision, power of vision. Okay, forget it. I don't need power. I can maintain my life like that. चलो जाने दो इसको. I am taking time. Do you know why? We have nothing to do in the lab. ये है matter behind this. We are not having microscopes. Without microscope now we are handicapped. We cannot go for clinical. We are less of
Oxygen supply.
means less of lack of energy production, lack of energy in the cells. Why lack? Lack of oxidation. With what I will be oxidizing? Oxygen will be required. So I am unable to extract oxygen from the atmosphere and supply this oxygen to the tissues. Then where is the problem? Hemoglobin. There is no sufficient hemoglobin. Why? Hemoglobin needs globin. Globin is a protein. You are not getting protein, you are not taking protein, so hemoglobin is not formed. Heme is there, globin is not there. So there is lack of production of hemoglobin. So what will happen to my body? So you see the importance of protein. My muscles, see the condition of my muscles, everything. Why? Getting atrophied because of aging. And your muscles are firm. It is hypertrophied. Number of muscle fiber cannot be increased. It cannot be increased. Then why it is becoming like that? After doing exercise, training? Yes. Size of the muscle, its muscle fiber is increased, has increased. We call it hypertrophy. Number of fiber is same, but their size is increasing. That's why this is increasing. मैंने रास्ता दिया था इधर से. There is a cut here. Injured. I injured my skin. Okay? So what will happen? After some time, some days, this part will be repaired. And for this repairing purpose, new cells has to be generated. So unless there is protein, can new cell be formed? So this part, this job also done by the proteins. There is no protein. So what is happening? Getting frequent cold, getting different type of infection. Means what? Immunity is reduced. Immune bodies are protein. Immune bodies are protein. My stomach, my belly is like that. Yeah? My skin, if I push like this, so there is a hole, it like depressed. That means what? Edema is there. Why there is edema? Because lack of plasma protein. Lack of plasma, low osmotic pressure. We have seen, if pressure is reduced from 25 millimeter mercury to 20, 15, what is happening? There will be more accumulation of fluid in the tissue spaces. So there will be edema. So these plasma proteins are protein. So I can give you a lot of energy, a lot of examples. The proteins are performing. These are not performed by any other nutrients. That's why protein is very important. Carbohydrate has got a protein sparing action. Means one of the function of carbohydrate is protein sparing. It spares the function of protein. It's protein sparing function. Means if I remain present, protein will be spared from production of energy. अरे भाई तुम्हारा vital काम है, तुम करो. Energy का purpose है ना, that I will do. But your job we can't do. You are driver. So we cannot utilize you for driving or pushing the car or doing other thing. Okay, you are a very important person. So that's why carbohydrates are called protein sparing nutrient. Okay. So anyway, let's come into this. Digestive function of pancreatic juice. This contains proteolytic enzymes. So we are eating food containing proteins. That proteins has to be digested into amino acids. Various kinds of proteins we are eating. Simple subprotein, conjugated protein, derived protein. Different kinds of protein we are eating. We have to digest. Elastin, as I noticed, gelatin. So that has to be digested. And this will be digested by the proteolytic enzymes. Can you name some proteolytic enzymes of pancreatic juice? One. Who will tell? Tell. Get up and tell. Chymotrypsinogen. He is wrong or correct?
It is
Why wrong?
inactive form.
It is the active form. In pancreatic juice, you will not find any active enzyme. I told you in my theory class. So these are secreted as inactive form, chymotrypsinogen. It only when it is released into the GI tract, into the intestinal, into the duodenal lumen, then only it will be activated by trypsin. These are not actually present there. These are inactive form. That's why I asked. You should not do this mistake. So pancreatic juice will not contain any. If they contain trypsin, it will digest its organ. Patient dies in acute pancreatitis. Immediately, acute pancreatitis patient may not die, may die. There is a chance. And no treatment, otherwise conservative treatment, only infusion. Severe pain. And pain is such that you will not find comfort in any position. This way, pain. This way, pain. This way, sleep. Pain. This way, sleep. You will not find any comfort in any position. You say pain is here. There will be back pain. And there will be fever, there will be nausea, there will be vomiting. When there is such pain, immediately hospitalization is required. And investigation to be started. First investigation, sonography or CT scan or MRI. What is the condition of the pancreas? They will say, it is having acute pancreatitis. Means there is inflammation in the pancreas. Means there are some, those enzymes which are preventing them, the secretions are unable to come out. So they are obstructed. Because of this obstruction, enzyme trypsin inhibitor, their activity, they are no longer, the function of trypsin inhibitor is, if by many means trypsinogen becomes trypsin, immediately trypsin inhibitor will inhibit it. But it has got some power, no? It can be overpowered by any situation in them. So during inflammation, its power becomes less. So there will be activation of trypsin. So the power of this trypsin inhibitor will be reduced. It is unable to stop. So there you will be active trypsin. They will start digesting. Chalo, pancreas khalo. ये तुम्हारा खाना है आज का. It knows only flesh. I got flesh, I got protein, and lipase, phospholipase, A2. It will split. It will also becomes activated. Lysolysis. Then it will become activated. It will also it is having power of necrotizing the cells. So there will be cell necrosis, killing of the cells. So there will be destruction of, and ultimately there are blood vessels. It will be affecting the blood. Immediately there will be profuse bleeding and hypovolemia. All the bleeding are in the abdominal cavity, in the peritoneal cavity. No blood to pump out. Everything came out of the blood vessels. Patient will die. So it's a very serious condition. That's why. No, don't destroy your pancreas. Okay? And next function, pancreatic juice. These are having carbohydrate splitting engine, amylase. With that, it can digest starch, soluble as well as insoluble. It can digest. If you eat liver, the liver will be having glycogen. You can eat liver, no? When you purchase mutton, no? So they give some portion of liver also. So some persons, I like liver. So within that liver there will be some glycogen. So that glycogen will be digested by. Yeah. Okay, so I am eating the tissues of mutton, of chicken. So there will be having elastin, collagen. So that will also be needed to be digested. So for that we have enzymes, elastase, collagenase, gelatinase we are having. So with that we will be digesting. Okay, and there are carboxypeptides. I already told. She is having the last amino acid, and this carboxylic group is free. So okay, I got. Yeah, the person having free carbohydrates. So cut it. Amino acids are free. So, and another enzyme is there. You will get it in intestine. It will start from that side. Free amino terminal, aminopeptides. Carboxypeptides will begin from that side. And so most of them are enteropeptides. Means in between. Peptide bond in between. Okay, cut it. So there will be soda peptides. दुखी हूं मैं, बहुत दुखी हूं. हाँ, मैं बहुत दुखी हूं. बोलो ना, एक बार बोलो, मैं दुखी हूं. Okay. Next function is, in this way we can there are some lipolytic enzymes. We have seen lipase, and co-lipase is helping the function of lipase, and how these are digested. I have shown this in the theory class. How lipids are there. Phospholipase, phospholipid can also be digested by phospholipase enzymes. Cholesterol esterase, with cholesterol esterase, we can digest cholesterol ester. Okay, in this way we can digest the fat also. So this way we can perform the digestive function. Neutralization action of pancreatic juice. When the acidic food enters from stomach into intestine, intestine cannot tolerate acids. Otherwise there will be duodenal ulcer. So it immediately should be neutralized. So when there is presence of acid, immediately there will be secretion of secretin. Now this secretin will stimulate the duct cells Hey, produce sodium ज़्यादा से ज़्यादा थी ready करो. So this duct will liberate. The pancreatic juice will be having lot of bicarbonate ions, and they will be entering into the lumen of duodenum, and it will be neutralizing them. So no ulceration. If sufficient bicarbonate comes, and for this we need what? Stimulation. Who will be stimulating? The acid itself. The acid present in the bolus. When they make contact with the wall is having some cells, eye cells, they will produce secretin. Now this secretin will be absorbed in blood, will come to the tubular cells. Okay, ductal cells. So there it will cause secretion of bicarbonate ions. More water. So with this lot of bicarbonate, they will be secreted into the duodenum. So with this alkaline substances, the pH will be alkaline. Acid will be neutralized. So second function we could know. And the third function, digestion of proteins we learn, digestion of lipid we learn, digestion of carbohydrates we learn, digestion of proteins we learn. Next, Kiri, the counter. Trypsin inhibitor, chymotrypsin. What is casein? When you drink milk, what happens is, after some time if you vomit it, what you will get? You get the milk or something else. So this is, yes, we call it chana. It's not curd. Casein. This is a casein. Calcium percassinate. Casein, it is done by the pepsin, active pepsin in stomach. So immediately this combines with calcium ion and deposited and precipitated. Okay, these are further digested by, when it enters into the intestine, they are digested by chymotrypsin and also by trypsin. So it is completed by actually chymotrypsin, digestion of casein protein. I talked about this. Unlike trypsin and chymotrypsin, these are exopeptides and split of specific carboxyl terminal amino acids from the protein molecule. What are these carboxyl peptides be? Okay. These are already discussed. Digestion of lipid discussed. Digestion, carbohydrate. Secretion of bicarbonate ions. From stomach, first food bolus came into duodenum. This is acidic. So immediately this acid is sensed by intestinal wall. There are some cells, eye cells. So they got stimulated. So what they secreted? They secreted secretin. Now this secretin will be absorbed in the blood and it will be circulated in the circulated throughout the body, and some also will be circulated into the pancreas. And in the pancreas, the ductal cells are there. So they are having receptors for secretin. So receptors will bind with the ductal cells. Okay, then it will start producing bicarbonate ion. How these bicarbonate ions are formed? Let us see. Question may come. This is the lumen. And this size, other ductal cells are there. So ductal cells, a lot of ductal cells are there. These are the lumen. Here also ductal cells. So this is the lumen. Okay. So this side is interstitial fluid. Okay, this fluid is there. So means blood is also there. Plasma is also there. Okay. So this side it is there. From the blood, carbon dioxide can enter through capillaries. Through capillary to extracellular fluid, from this extracellular fluid to it may come to duct cells. Okay, now you see what happens. This carbon dioxide enters here, and this cell is having lot of carbonic anhydrides. So immediately it combines with water and forms H2CO3. This H2CO3 is dissociated into bicarbonate ion and hydrogen ion. This bicarbonate ion enters into the lumen, into the ductal lumen. And in exchange, ductal lumen is having chloride ion. In exchange, it is entering within the duct cells. So this is what? Chloride bicarbonate exchanger. So this is main, and it requires energy. It requires energy, means breakdown of ATP. So with this, with the help of this, the bicarbonate produced here is eliminated to the lumen. And in exchange, chloride ion is entering from the lumen into the duct cells. Again, this chloride ion is coming back to the duct. With the help of CFTR controlling chloride channel. What is CFTR? Cystic fibrosis transmembrane regulator. Cystic fibrosis is a dangerous disease. You will find such children always running nose and a lot of sputum, thick sputum with bad smell, with pus cell in it may come. Means what? There may be sinusitis, means inflammation of the sinus. Blockage of airways. Why blockage of airways? Airways means respiratory airways. Why? Just think. This is our trachea, and it will be divided into two. One will be coming here, another will be going there. Okay? Then again they will be dividing. They will be dividing. A lot of divisions, then alveoli will be formed like grapes. Okay. So now this tract are called airways. If we take a section of this tract, excluding this will be having a cartilage ring, C-shaped ring. C-shaped ring, and this open size is here because here the digestive tract is going side by side. So that's why it is there. And inside, if you get a section, and you will find inside is lined by a epithelial layer of tissues. And this epithelial layer of tissues this single layer of epithelium, like this. Okay, single layer of epithelium. Some are mucus cells, some are non-mucus cells, but having cilia. Means there is even a lot of ciliated epithelium. And these cilia are performing certain functions, very important functions. See, this tract, the epithelium, constantly producing mucus. Why? It is making a coat. Why? You are inhaling air, it may be dry, like this season. So while it is passing through, it may take up water vapor from these cells. These cells may dry and may become dead. So to prevent this, so that the cells are not dried by the passage of air, so there should be a coating of mucus. Moreover, why we inhale? Our nose is having hair. What is the purpose of this hair here? Huh? अरे मुझसे न घुस जाए और flies, they may enter. Okay? So to prevent them, we need it. This is the purpose. And you see, inside of this is red. Why it is so red? There are a lot of capillaries. Why? The capillaries are here. If something happens, immediately there is nasal bleeding. Because these capillaries are almost at the surface. Almost at the surface. Why? Because the air we will be inhaling, say in this season, is very cold. Is very cold. So this air, before entering into the alveoli, it should be warm and become 100% what? What having 100% water vapor. It should be saturated with water vapor. Unless, while going through, it will extract water vapor. So that cell may die. While passing through the terminal branches, there you will not find any cilia. There you may not find any mucus. So what will happen then? They may destroy those cells. So before that, I have to make them humidified. I have to make them warm. See, when oxygen is given to a patient, so before entering to the nose, it runs through a container having water. Why? It is humidifying there. Then only it is passed. So this is needed. For that purpose, this is very, red. Okay. So now, with this air, we can inhale. So while you will be passing through this road, there are a lot of dusts. A lot of car is moving and a lot of dust will be moving in the air, floating in the air, and you are inhaling. Some person may use take out their mask and put it there, so that even then some dust will be coming. This may damage the functions. Okay, so we have to protect our lung from this dust. So they will be trapped by the mucus. So this is another important function. Bacteria, virus, they will be trapped there. They will be trapped there. And, okay, mucus is there, they are trapped. Now, what will happen if this mucus with dust, with a lot of these things, it stays there, what will happen? The bacteria says, "Hey, this is nice place to live. Okay, let's live here and start producing new cells." Means site of infection, site of inflammation I have created. So what can be done so that it doesn't happen? Eliminate it. Generate new mucus. The old mucus, eliminate. How to eliminate? With the cilia. Go out. So this cilia will move like this. So all mucus will be coming. Then when it comes here, and it will irritate the cells there and will generate reflexly cough. And with this cough, she took out some phlegm from mucus from there. And what she did? No, not done. She swallowed it. You are also doing it. You are also doing it. Because here is the larynx. So when it will be coming here, so there you will find a cavity there. So it will be what you will do? It will coming to the GI tract. Okay, now you go. Okay, and if you are able to cough, then you will be eliminated this. So this is the process. So if it is healthy, so always I will be producing mucus, and it will be, my cilia will be helping to eliminate the older mucus to the throat or to eliminate through by cough. Okay. Now, I am a smoker. Oh, what's up? Now I am able to concentrate in my studies. It was very difficult, no? Now it can be solved easily. Because this cells now is activated. Buddhi, aa gaya. So that's why somebody smokes. And if it is done day after day, okay, it's like, somebody who starts smoking, suppose you will be starting smoking, you will be, okay. So you will not go to shop, give me one cigarette, no. She smokes. Okay, she will be telling you, smoke, no? See, you'll find moja. So she has offered you, you initially denied, then you took, then okay. Again it started. She offered free of cost. Then okay, every time she is offering something, and after that, what will happen? You will go to shop. She is not there. Okay, give me one cigarette. It started. 1 cigarette, 2 cigarettes, 3 cigarettes, 1 packet. She became a smoker. Then what will happen? Lot of things can happen in your body. It doesn't spare any organ of my body. It doesn't spare. First, gradually it will paralyze the cilia of ciliated epithelia. So, you will be unable to eliminate them. As a result, smokers are always we call it smoker's cough. Nothing is coming out. This is sticky, no? It's there. Not fresh mucus is not generating. It is written there. That's why it is trying to eliminate. It increases cholesterol level. When you do it, no? So lot of carbon monoxide is produced. And this carbon monoxide, when entered into the blood, from lung diffuses into the blood, it will form a permanent compound with hemoglobin. And it means it is occupying the bond by which actually it previously used to carry oxygen. Now this is occupied by this valency is occupied by the carbon monoxide. So you made this hemoglobin useless for you. So lot of injurious effects of smoking is there. You understood this? No, not yet explained. So this carbon dioxide combines with this, with the help of carbonate is converted into H2CO3. Then this bicarbonate is formed. It is liberated here. In exchange, chloride ion is entered. And it was the topic. What is the topic? What is this? Cystic fibrosis. It happens in breathing. This airways become clogged. Why this is clogged? Because of accumulation of this. Lot of mucus will be formed and they will be clogged. As a result, unable to breathe. There will be a breathing problem. Similarly, it may happen in pancreas. How? There will be a lot of secretion of mucin and bicarbonates. And they may clog the duct. So, this secretion will not get passage to enter into the, because of if this chloride ion is not available. See, if this chloride ion is not available for this, there will not be any secretion H2CO3. So, no alkali. Only the enzymes, mucin-rich secretion will be there, and they are viscous, not watery. This, along with water, make it watery. But since this is not formed because of absence of this, this is not formed, means it will be less watery, means it will be thick, viscous. So this viscous thing, while passing through the duct, will clog this, will block this passage. So if it is blocked, then the pancreatic juice will be unable to enter into the lumen of the duodenum. As a result, what will happen? Gradually, the pancreatic acinas will stop secreting enzymes also. Since there is no movement of juice and there will be non-functioning of the acinas. And it happens this is called pancreatic insufficiency. So, this pancreatic insufficiency occurs due to absence of this. If this is not present, this will not be done. If this is not, the bile will not be alkaline, it will not be bile. Pancreatic juice will not be watery. So there will be thick pancreatic juice formation, viscous way. So this may clog the duct, this may block the duct, and as a result, the kidneys may die. Okay, so because of that, there will be insufficiency. Means pancreas exocrine function will not be able to do its job. And it happens in cystic fibrosis. This is inborn disease. Okay. these children, they are having trouble in, there will be this sort of, so wherever mucus is produced, mucus producing organs, they will suffer this. So it happens in cystic fibrosis, very bad disease. Okay, now see what will happen to this hydrogen ion. What will happen to this hydrogen? So there will be a sodium hydrogen exchanger. So this hydrogen will be coming out with a proton, but the cell is then deficient of one proton, so it will be exchanged with this. So sodium will be coming in and hydrogen will be coming. But the sodium will not be allowed here, that will be sent back to the plasma, sent by this pump. In exchange, potassium ion will be entering. Okay, so this hydrogen will be coming out, sodium will enter. This sodium ion with this pump will be coming out and potassium ion will be entering. This is the process by which bicarbonate is formed. Can you understand this? Okay, you will find it in, what is this book? Not Ekejen, G.K. Pal. Same thing? Mechanism of pancreatic secretion. This is, see, the Echinus cells. See the Echinus cells, what they are doing. This is the Echinus cells. The bulb-like structure. So there are cells, these are Echinus cells. So this will be the apical side. This is the base. And there are a lot of Echinus cells in this way, and the duct is coming out. So here at the base, see, there are a lot of receptors. One receptor is for GRP, gastrin releasing peptide. They activate this, when they bind this is the local hormone secreted from the nerve endings, and they will, when they bind with this receptor, they will stimulate release of calcium ion. They will cause release of calcium. Acetylcholine liberated from the vagal nerve endings. We know vagus nerve stimulate production, pancreatic juice formation. So there will be lot secretion of calcium ion. There will be secretion of calcium ion. Gastrin, CCK, cholecystokinin, pancreasamine. Okay, so secretin, gastrin and CCK, they will also, there are receptors, they will also stimulate this cell by modifying these receptors, and their second messenger is calcium ion. So all this will produce the increased level of calcium ion, and this increased level of calcium ion will help exocytosis of vesicles having zymogen granules having lot of enzymes. So within the packet, what is there? Different type of enzymes. So they will be exocytosed into the acinar lumen. Okay. And this is secretin. This secretin is liberated from intestinal because of contact with acidic substance. Eye cells are there on the lumen. So when they made contact with the acidic cells, hydrochloric acid make contact with, they produce secretin. Now this secretin will be circulated, and through circulation it will reach here and it will stimulate this, and it will increase cyclic AMP as second messenger, and this will increase, this will also do the same job like this, and there will be, so in this way the different enzymes are secreted in the pancreatic juice. So previously we have seen how bicarbonate are synthesized and released. Here we have seen how the zymogen granules, this means these enzymes are synthesized and released. So there are two mechanisms. One is humoral mechanism. This is humoral mechanism. And neural mechanism is, I will meet after going there, attending this class, I will eat meat, chicken. So you are thinking. So what is happening? So this thinking process is coming from cerebral cortex, from amygdala, from hypothalamus, satiety, appetite center, will coming down to medulla. There is vagus, nucleus of vagus. So they will be stimulated by the nervous system, central nervous system, they will be stimulated. So stimulation of vagus nucleus will cause activation of vagus nerve. Now the vagus nerve, as 10th cranial nerve, will travel. And it will come to a ganglia. And that ganglia is located in the pancreas. So this will end in the ganglia. From this ganglia, postsynaptic neuron, postganglionic fiber will be generated. And this fiber will be ending here. And this ending will liberate acetylcholine. And this acetylcholine will bind with this and increase the calcium level, and it will cause this. See, my thinking process can also cause secretion of peptic juice. Acinar juice. Just thinking. These are called psychic stimuli. Even the presence, when the food will be entering into the duodenum, wall will be distended. So some stress receptors are there. These stress receptors will be stimulated, and this stimulation will go to medulla, vagus nerve will be stimulated, and this will, while ending into the ganglia, they will make two branches. One branch will liberate acetyl coenzyme A, another branch will liberate GRP. And this GRP will bind with that and it will produce calcium ion. It happens in cephalic phase. This is the mechanism of cephalic phase by which pancreatic juice secretion is made. There is another intestinal phase. So intestinal phase starts when food enters within intestine. So it starts with acid. It starts with acid. It starts with distension of the stomach or intestine. So one stimuli is distension, another stimuli is acid. So acid will cause, acid has to be neutralized. So it will cause secretion of secretin, and the function of secretin is to increase the bicarbonate biosynthesis. And there will be some products of protein digestion that has been done by the in stomach. Some pepsin has been formed. Now this pepsin, presence of this pepsin in the duodenum will also stimulate another receptors. They will cause secretion of GRP. As a result, what will happen? The cell, the acinar cell will start producing enzymes. So, end products of protein metabolism, presence of end products of protein metabolism, fat metabolism, they will initiate local reflex. They will initiate synthesis of local hormones. So, these local reflex and local hormones will also cause stimulation of the acinar cells as well as stimulation of the cells of the ducts. So, by that, bicarbonate production will be increased. By that, enzyme production will also be increased. So, these are the three phases. One is cephalic phase, one is gastric phase, another is intestinal phase. You will find this very simple. See, what is the path? Initially is cephalic phase. Sight, smell, taste. These are what? These are the psychic stimuli plus chewing. They are called vagal stimulation. Vagal stimulation through acetylcholine will cause pancreatic secretion rich in enzymes. Okay, now this will digest. When these are digested, there will be inhibition of secretion of all those things. No need. No, means everything is digested, then what is the need? So there will be inhibited. Now this is the cephalic phase. Now see the gastric phase. Here the peptide and amino acids will be formed by the action of the pepsin. So with this, the food will be entering within the stomach, from stomach within the intestine. From there, gastrin will be produced because intestinal wall also produce to some extent of normally gastrin is the product of hormone of stomach, but there are some cells in the intestinal wall, they also produce gastrin. Now this gastrin will stimulate this and it will produce these enzymes. And this is the gastric phase. Now intestinal phase. Now see, from this it has entered into intestine. Entered into intestine. Acidic chyme, so it will cause S cells or argentaphin cells, same name, to produce secretin. Now this secretin will cause pancreatic rich in bicarbonate ion. Okay, now acid will be neutralized. So this is in this way, this mechanism. This is the mechanism, okay? It's almost finished. This is summarized. Last part. What is written there? Read. Ghrelin is secreted from stomach wall when it is empty. Ghrelin stimulates brain to produce hunger. When is our stomach empty? We feel hungry. It is because the stomach wall will produce ghrelin and it will stimulate the brain, hypothalamus, there is hunger center. And this hunger center is not only stimulated by the ghrelin, there is another. When you came, you only. You were there from the beginning. No, when you came. Oh, you are from beginning. Okay. When we feel hungry, when our blood sugar level falls, we hear hypoglycemia. When this hypoglycemic blood enters into hypothalamus, it says, sorry, there is less glucose. So, blood needs more glucose. What has to be done? So, I stimulate the hunger center. So, I started feeling hungry. Okay, khana do, kuch bhi. So, when you will be eating, your stomach will be distended. So, there will be on a reflex path. My stomach is reflected, there will be distension, there will be stretching of this reflex, they will be stimulated. Now it will go to the organ center, it will stop. Stop, no more hunger. I am saying now satiety has been made. So no more food is required. Okay, so I am now, okay. Feeling? What happens when you take a lot of food thereafter? One feeling is there. What is this called? Satiety. Feeling of satiety will come. With the distension, when there is, food is not there, you are feeling hungry. So mechanically I distended your stomach. You will feel satiety. When our stomach is distended because of gas or something else, we don't feel like eating. Because of stretching of this, inhibition of the vagal center, the appetite center. So that's why you don't feel like eating anything. Let's see what can be done.
All of you know this is the RBC pipette. You will take the blood, first you will draw the blood by sucking it with your mouth. we will draw up to this 0.5 mark. And if it is more, if it is getting more, then draw the blood like this.
After taking little bit more, you will wipe the extra blood that is adhering to the tube from outside.
So how will you know that
Hello.
which one is the 0.5 mark? There are two
Hello.
marks you can see, na? It is written 0.5, and here also is one mark, and here also is another mark. So which mark will be the 0.5 mark? Middle? There is no middle mark. Yeah, lower one will be the 0.5 mark. So you have seen that in this case the blood have gone upwards more than the 0.5 mark. So if the blood is going upwards, then we will bring it down by using a tissue paper. We will just tap it. We will just hold the tissue paper here, and we will just use it like a blotting paper. Then the blood will come down. The level will come down by itself. Tissue paper.
টিপে যে এক্সট্রা ব্লাড লেগে আছে বাইরের দিকে ওয়াইপ ইট
Okay, firstly we will wipe the tip so that we don't get confused by the level of the blood.
if is see what happens if this extra blood is retained on the surface you will be dipping it into the RBC diluting fluid, so that blood will be mixed with the RBC diluting fluid. Somebody else will be using this, some RBCs will be entering through this fluid, so count will be different. So you first to wipe this extra blood.
So we will drop the amount of blood which has crossed the 0.5 mark. After that we will keep the pipette horizontally. If we keep it horizontally, then the blood will not drop. If you are keeping it vertically, then due to gravity the blood level will come even further. So after taking this much of blood, we will then draw the diluting fluid into the pipette.
So blood has been taken up to 0.5. Then we will take fluid up to 101. So by this process, blood will be diluted. How many times? 0.5 to 101. 200 times. Okay. It will be blood will be diluted. Remember this. It is diluted. So when I will be calculating RBC count, initially it will be the count of diluted blood. So since it is diluted 20 times, so I have to take it to the original value. So divide by multiplying by the value, multiplying the value we obtained, then we have to multiply it by 200, then the value will be actual blood. Count for actual blood per cubic millimeter.
So after taking the blood, I have taken the diluting fluid up to the 101 mark. So here you can see that it has crossed the 101 mark. So if it has crossed the 101 mark, as I have shown you before, in the similar way, by using a blotting paper or a tissue paper, you will bring the blood down, bring the fluid down. We will not mix it. If it has gone above 101, firstly we will not mix it. After it comes to this level, 101 level, then only we will mix the blood. So you have seen that in this case it has crossed the 101 mark. So in the similar way, and we will keep the tip closed by
Now
pointing.
this will not be, anything will not be coming out.
Yeah. So air pressure is not there. It is vacuum is created. That's why there will be no dripping of the blood. So we will either put our finger here like this and stop the flow of air, or we can bend the tube like this so that there is no further flow of the blood or the fluid. Then we will use the blotting paper to bring the level down.
And using blotting paper is a little bit risky, tissue paper also. If you keep it just touching for a long period, then it will suck all the fluids. So just touch it and remove it. Touch it, remove it.
If it is not coming down by gravity, you can gently blow it so that the level comes down. So after this level comes down, we will keep this pipette horizontally in a table, or we will just keep it somewhere in a horizontal position and then we will mix it.
ব্লু করে কন্ট্রোল করতে পারবে না।
We will mix it for 2 minutes
ব্লু করে করব একদম
so that the
টাচ করে
fluid
করতে হবে।
gets
ব্লু করলে
mixed.
একদম নিচে চলে আসবে। আবার এটাকে ডিসকার্ড করে ফেলতে হবে। কন্ট্রোল তুমি দ্যাট মাস কন্ট্রোল ইউ ক্যান ডু।
So suppose I have mixed it for 2 minutes.
No blowing.
After that,
Only touching.
we will discard 2 to 3
Okay.
drops of blood, so that the amount of fluid which has been mixed with the blood does not dilute the fluid, dilute the blood count. So the blood count will be less. So the fluid which has not been mixed with the blood, we will discard that much fluid. Suppose here we will discard 2 to 3 drops of fluid. We can mix like this by holding it like this. We will mix it in our hand like this for 2 minutes by keeping it in a horizontal position.
এটা ছেড়ে দাও, এটা বেরোবে না, এটা ছেড়ে দাও। হ্যাঁ হ্যাঁ হ্যাঁ। While you will be doing, bring some rubber band. So by folding it, then you fix it. Okay, so there it will be airtight. That's it. So no fluid will come out. Okay, so when you will be using the fluid, then you remove this girdle.
Okay, suppose we have mixed it for 2 minutes. After that, we will discard 2 to 3 drops of this fluid.
So you have to drop 2 to 3 drops.
What will happen if you take the first drop for charging? You won't find any RBC there. Because this portion, there was no mixing of blood. Okay, that's why I have discarded.
Okay, after discarding these 3 drops, now there is only fluid which is mixed with the blood. So we will take a hanging drop. We will take a drop in such a way that the drop hangs.
See, one
from
drop
this
is
pipette.
coming. So this has to be touched.
These drops will be
Yeah.
hanging from the pipette. And when the drop is hanging from the pipette, we will gently touch the water droplet. Only the water droplet we will touch.
Wait. See, both ends, there is some. No, again, not about. Not talking about gap. And we will come enter into this.
Under behind, they cannot see maybe.
Sure. No?
And I'm holding this. Okay. Then we will touch this hanging drop in such a way that it charges the slide. So you can see
it is.
that the water droplet is spread beyond under this cover slip. This is called
excess-
charging.
Excess fluid is drained. Okay, there is some fluid here. So how to remove this? Just touch it from outside. Let us go. Bas. Okay, this side is charged. Now he will charge this side also.
In the similar way, we will take a hanging drop.
Down, touch. 45-degree angle. Bas, hey, going, bas. It is already spread. You will be seeing it. This is going. Okay. Bas, over.
During charging, we will maintain a 45-degree angle with this pipette like this.
So nicely, it is. Will you be able to do it? Okay. Run.